Welcome to Nusantara, the Southeast Asian archipelago. It is the birthplace of humanity, in our long journey of civilization. A homeland for human ancestors, for the last 1.5 million years. A place where our early ancestors have roamed a vast land of opportunity. Shared the space with many other wonderful creatures. Leave us the evidence of human evolution and their adaptation to the environment. Who are they? What do they look like? And how do they live their life? Our mission is to provide best practices in the work of archaeological research and development. Pushing the values of our research beyond the knowledge. Reveals the national identity. Enriches our understanding of cultural developments. Unveil the reason behind our strong character of diversity. Narrates the story of ethnogenesis and the foundation of our nation state. Preserving our past as a heritage for the wealth of our future generation.
Selamat siang, Bapak Ibu sekalian. Selamat datang di acara talkshow CPAS ke-22 di siang hari ini, di tanggal 23 Januari 2024. Kami dari CPAS mengucapkan semoga belum telat, selamat tahun baru, dan kita akan mengadakan diskusi sebagai diskusi rutinitas bulanan dari CPAS untuk mendiseminasikan hasil riset dari para peneliti di seluruh dunia, di dunia arkeologi kepada masyarakat. Dan tanpa menyita waktu lebih lama lagi, seperti biasa, kita akan mendapatkan sedikit pengantar dari Profesor Truman Simanjuntak selaku Ketua dari CPAS. Halo, selamat siang, Prof.
Selamat siang, Mas Ruli.
Sehat, Prof, ya?
Sehat.
Prof, selalu sehat. Terima kasih, silakan Prof. Monggo untuk pengantar pada diskusi kita siang hari ini tentang paleontologi di Indonesia.
Baik, terima kasih. Selamat siang buat semuanya. Ya, sudah menyempatkan hadir di dalam talkshow kita kali ini. Dan ini sudah yang ke-22, ya, ke-22. Dan kita selalu setia menyelenggarakan talkshow itu setiap bulannya. Ya, selamat siang buat Pak Gert ya. Ini tamu kita istimewa untuk talkshow kali ini. Dan
selamat siang, Pak.
Ya, selamat siang Pak, dan juga ada tamu-tamu khusus kita, yaitu Mas Unggul. Mudah-mudahan, saya belum lihat, mudah-mudahan sudah on time juga ada di sini. Dan tentu
Siap.
juga rekan-rekan dari CPAS, Center for Prehistory and Austronesian Studies, yang telah mempersiapkan acara ini hingga kita berharap bisa berjalan lancar sampai akhirnya nanti. Mohon maaf kalau saya agak terlambat dan juga kelihatannya lain dari tempat saya tidak begitu lancar. Dan kebetulan mulai jam 1 ini saya memberikan kuliah, jadi saya hanya mencuri waktu sebentar untuk ketemu para peserta termasuk Pak Gert ya terutama dan teman-teman lainnya untuk bisa berjumpa dan setelah itu nanti saya akan meninggalkan talkshow kita dan saya berharap pertemuan ini, diskusi-diskusi yang akan berlangsung nanti bisa berjalan bagus dengan munculnya ide-ide ataupun pertanyaan-pertanyaan yang menarik di seputar topik yang akan kita perbincangkan. Jadi kita beruntung di siang hari ini mendapat tamu istimewa yang menjadi narasumber kita, yaitu saya kira sebagian besar dari kita sudah mengenalnya. Paling kalau ada yang beberapa yang belum, ya di kesempatan ini saya ingin memperkenalkan sedikit. Jadi kalau bicara tentang paleontologi Indonesia, ya beliau inilah yang terdepan. Ya, namanya Pak Gert, panggilan akrabnya, nama lengkapnya Gert van den Bergh. Ya. Jadi dalam kesempatan ini kita coba pilihkan sebuah topik atau isu yang sangat menarik dan tidak akan pernah usang untuk diperbincangkan, yaitu mengenai Biostratigraphy and Biogeography of Quaternary Vertebrates in Indonesian Archipelago. Pak Gert akan menceritakan kepada kita ya, seperti apa gambaran dunia hewan pada era kuaterner di Indonesia, ya. Seperti apa keragamannya, ya, dan bagaimana kira-kira persebarannya dalam ruang dan waktu hingga kita memperoleh tinggalan-tinggalan berupa fosil dalam endapan-endapan tanah atau dalam lapisan-lapisan tanah. Nah, ini di sini pentingnya mempelajari biostratigrafi ini untuk mengetahui bagaimana keberadaan hewan-hewan itu dan bagaimana pula evolusinya dalam rentang waktu jutaan tahun. Dan tentu ini sangat mengait dengan biogeografi, dengan lingkungan di mana hewan itu hidup. Ya, seperti apa hubungan antara ya, kondisi geografi dengan kehidupan atau keberadaan hewan-hewan itu, dan seperti apa pula ya hubungan antara kondisi geografi dengan kehadiran dan kedatangannya ke Kepulauan Nusantara kita. Ini isu yang sangat-sangat menarik dan kita berupaya ya untuk memahaminya lewat ya penyelenggaraan talkshow ini. Jadi mari kita manfaatkan sebaik-baiknya kehadiran pakar kita, super pakar kita, yang akan menjelaskan kepada kita tentang gambaran biostratigrafi dan biogeografi kepulauan kita. Sekali lagi terima kasih kepada Pak Gert, kepada Mas Unggul, kepada rekan-rekan member CPAS yang sudah mempersiapkan, dan tentu saja kepada semua partisipan yang tidak pernah lelah untuk bergabung dengan kita. Ya, kami akan selalu berupaya menyelenggarakan event-event semacam ini yang tujuannya mencerdaskan kita. Lebih-lebih kalau partisipan semangat untuk hadir. Ya, saya lihat ada di sini Pak Iskandar juga. Terima kasih Pak Iskandar
Selamat siang,
menyempatkan
Pak. Selamat
hadir.
siang, Pak.
Ya,
Makasih, Pak.
bukan main Pak, bukan main. Terima kasih sekali menyempatkan hadir di dalam talk show ini. Dan tentu masih banyak yang lain yang saya tidak sebutkan satu persatu. Sekali lagi terima kasih kepada kita semua yang ada di sini dan selamat berdiskusi. Begitu, Mas Ruli.
Baik, terima kasih Prof. Wah, senang sekali ya Prof. Kita kali ini kedatangan pakarnya, Pak Gert, yang sudah lama sekali saya baca banyak sekali artikel beliau. Apresiasi sekali lagi. Thank you very much Gert for being here in the 22nd talk show for CPAS, Center for Prehistory and Austronesian Studies. Thank you Prof. Truman atas pengantarnya, beautiful introduction. And then I will invite Mas Ami as a moderator today. Hello Mas Ami.
Halo.
Mas Ami,
Halo.
halo Mas Ami adalah anggota dari CPAS juga. Beliau sekarang menjabat sebagai dosen di Fakultas Interdisiplin UKSW Salatiga. Dan kepakaran Mas Ami tentunya di bidang zooarkeologi dan paleontologi vertebrata. Jadi apa kabar Mas Ami?
Baik.
Semoga sehat dan selamat menikmati diskusi ini dengan dipandu oleh Mas Ami. Mas Ami, waktu dan tempat saya persilakan.
Terima kasih banyak, Mas Ruli. Terima kasih banyak atas pengantarnya, Pak Truman, dan seluruh hadirin. Dan tentunya terima kasih sudah bersama kita, Dr. Van den Bergh. Saya one of your big fan. So, I think I don't know if I should introduce him in Bahasa Indonesia or in English. So I will just do it in English. So good afternoon, everyone. And allow me to introduce our guest today. He is a very, Van den Bergh is a senior lecturer at the Faculty of Science, Medicine and Health at the University of Wollongong, Australia. And he has this very profound expertise in vertebrate paleontology, zooarchaeology, geoarchaeology, and he is a leading authority in the field of quaternary vertebrate with a primary focus on the Indonesian archipelago. His research delved deep into the intricate realm of biostratigraphy and biogeography, unraveling the secret of the Earth's past. Today, he is kindly agreed to share with us his expertise on our captivating topic, biostratigraphy and biogeography of quaternary vertebrate in the Indonesian archipelago. So without further ado, over to you, sir, Mr. Bergh.
Oke, terima kasih. Saya minta maaf karena barusan ternyata kamera laptop saya tidak berfungsi, jadi ada hitam. So saya matikan lagi karena percuma kalau dinyalakan. Tapi saya harap bisa dengar suara dengan baik. Kalau begitu, ya. Terima kasih banyak untuk mengundang saya untuk hadir ke talk show ini. Yeah, let's just start. So I'll share my screen. So can you see that? Can you
Yes.
see an elephant in the room? Okay. Okay, so this thing here. Okay. So the title, Biostratigraphy and Biogeography of the Quaternary vertebrates in the Indonesian archipelago. First of all, I have to excuse myself because there was a bit of a misunderstanding. Initially, I thought that I had to talk on the biostratigraphy and biogeography of the island of Java. But then I saw in the invitation Indonesian archipelago. So I have, at the end of the presentation, I have added some slides on Sulawesi and Flores. Yeah, I also hope that I have to say I couldn't find all the images that I wanted because I have a lot of material that I don't have on my laptop here at my disposal. So I had to improvise a little bit. So I hope you don't mind. So let's just start with the first slide. Okay. So yeah, as you all know, I'm not going to stand still on this very long time, but terrestrial fossil faunas, they can inform us about a lot of things, provided that they are collected or excavated in a tight context. So that, and that's a bit, as we will see, the problem with the early excavations of Dubois and von Koeniswald, that there is just a lot of question marks. And in those days, the researchers, they didn't have a lot of competition from other people working in the same field, so they could basically tell what their thoughts were, and that was then becoming gradually a kind of paradigm, and without much critical thought on these things. So I will come back to that a bit later. But of course, fossil faunas of terrestrial vertebrates, they can provide us an age, a relative age of the geological layers that they are found in. Now, I must directly admit that large vertebrates like elephants or bovids, they are not that good for assessing the age of deposits. Moreover, we also have to take into account paleo-environmental factors that influence the composition of a fauna. And yeah, in modern research, we tend to take those variables more into account. But the problem with large megafauna is that you don't often find them in large quantities in a single layer. There are some exceptions, of course, like Trinil or Nandong, or on Flores, where I do my most of my research at the moment. But a lot of fossil finds, they are isolated finds, sometimes from the surface. So for assessing the age of geological strata, for biostratigraphy, they are not so good. And another thing is that nobody or microfossils of rodents and small mammals, they have hardly been studied yet in this region. And they, because their rapid turnover rate, they evolve faster than, for instance, elephants. So rats have much faster evolution. So evolutionary change in small mammals goes much faster than in large mammals. So they would be more suitable for making a biostratigraphy, similar as in marine layers, where we also use microfossils, of which there are usually heaps, like foraminifera. But yeah, fossil faunas can inform us in certain cases about the paleoclimatic or environmental conditions or changes in these conditions. One of the examples is, for instance, if you find a fossil of an orangutan then you can be pretty sure that there was a rainforest at the time when that layer was formed. Of course, paleogeography is another important aspect of why we study fossil faunas. Human subsistence patterns, and of course, as you all know, in Java, Homo erectus has been around for about 1.5 million years, but we have to admit that we not really know a lot yet about how Homo erectus interacted with fauna. And I guess there's still a lot of work to be done there. And then, of course, a question that many people ask is, why did all these large, or a lot of large animals become extinct during the Quaternary? And that is a question that a lot of paleontologists, also in Australia, but also in Indonesia and in the American continent, try to answer. And it's very difficult to prove that, for instance, an extinction is caused by hominins or by, for instance, climate change. And that is, of course, because the fossil record is not complete. But before, let's stop with all the restrictions that we face as paleontologists and see what we can do that we are better in. So, as you all know, quaternary natural history research is a multidisciplinary branch of research. It involves archaeological research in case there are, if we talk about the Quaternary, we know that ancient hominins were around. But in order to understand why fossils accumulate at a certain place, you need to know a bit about sedimentology, about how certain strata are formed, were they formed in a sheet flood or a major flooding, or in just a meandering river system, those kind of things. And they can then inform you a bit better on how fossils accumulated. If they are associated with volcanic deposits, maybe there was some volcanic hazard that caused the mass death of fauna, and I'll show you some examples from floras later on. Then, of course, paleontology itself, you have to classify what kind of fossils, what kind of animal, what taxa they belong to, what are their relationships with taxa elsewhere. Palynology, preferably, should also be used in combination with paleontologies to get a better idea of the flora or the vegetation and the climate at a certain time period. And then taphonomy is also a very important aspect. It can inform us a lot about how bones accumulated in the first place. Was it just an accumulation over a long period of time, gradually with bits of animal remains accumulating, or was it a mass death event? Those kind of things. Were the bones transported, or were they close to their original place where the animals died? And then, of course, geochronology, very important, and that's what we now have, what people like Dubois or from Koningswald didn't have. They didn't have ways of absolute measures of time, but now there's a wealth of data and methods, dating methods available that we can try to apply. And especially in a volcanic region as Indonesia, volcanic ash layers are really suitable often for getting a number on the age of a certain layer in which fossils are found. You can, of course, also try to directly date fossils now with uranium series dating. So there's a whole range of dating methods available. Optical dating, just to mention a few. And then internationally, ancient DNA extracted from fossils is a big topic. Unfortunately, in the tropics, that would be a bit more complicated because DNA doesn't preserve very well under hot, wet, humid conditions. But maybe the last couple of thousand years in archaeological sites, that's could be an important method. And also other geochemical methods and such as stable isotopes. Okay, so starting with Eugène Dubois, I pass Raden Saleh here, because Eugène Dubois was the first person actually to systematically excavate on a large scale. So he collected an enormous collection of fossils. And there we come directly at the problem in the modern times. If you would like to do an excavation like Dubois did, then that would cost a hell of money. So it's very difficult to repeat those kinds of excavations. So the Dubois collection of the fossils that he excavated at Trinil and other places in Java and also Sumatra is still a very important collection or a source of knowledge about the faunas of the past in Indonesia, including Homo erectus, of course. I won't mention too much Homo erectus fossils. Problem always with Homo erectus is that you have a very small sample, whereas in some animals you have a much wider sample, and you can do more about in interspecies variability. For instance, if you have a lot of bovid teeth, you can do some statistics on that, whereas on isolated hominin finds, that's always a problem. Okay, then another important figure after Eugène Dubois was, of course, Gustav Heinrich Ralph von Koenigswald. And I plucked this image, I think, from the Sangiran website. I didn't. Yeah, von Koenigswald, he came to Indonesia in 1931 as an employee of the Geological Survey of the Netherlands Indies. And he started to make sense of the fossils, the fossil vertebrates that were collected. There were excavations being carried out, for instance, at Nandong or in the Bumyayu area by the survey. And his task was to analyze these vertebrate remains and try to make sense about the fauna succession. And what he came up with in 1934 and 1935 was the fauna succession that you see here to the right. So the youngest fauna was what he called the Sampung fauna, based on a fossil assemblage from an archaeological site, Sampung cave site. Then the Ngandong fauna, based on the contents of the Ngandong terrace along the Solo River. And then older Middle Pleistocene, he estimated, was the Trinil fauna. And von Koenigswald basically correlated the Trinil fauna with fauna elements that were collected by Dubois in Quaternary. So according to the scheme of von Koenigswald, the Trinil fauna contains both elements from the excavations at Trinil, but also from Quaternary, and also from a number of other sites. And then the same with the Getis fauna, which he considered as slightly older than the Trinil fauna, early Pleistocene. And then based on excavations that the survey had conducted near Bumyayu, he had a Kali-Glaga fauna, which he considered to be early Pleistocene, and then a Chijulang fauna, middle Pliocene, sorry. And then the Chisande fauna, which was basically based on a single molar of a rhino. That was the Chisande fauna, early Pliocene or even Miocene. Now, the problem with this scheme was that, as I already briefly mentioned, a lot of these faunal assemblages, they were a mixture of fossil assemblages from different places. And after a while, it became clear that this scheme could no longer be held. And when I first, I have to also mention maybe Dirk Albert Hoyer, who was curator at of the Dubois collection in Leiden from 1946 to 1979. And he described a lot of the fossils, because one thing that Dubois himself had neglected to do, or had no time for, or didn't want to, was to sit down and analyze the fossils that he had collected and describe them. So his focus was primarily on the Homo erectus fossil and the fauna. He never published a decent paper on the fauna succession with detailed descriptions of the animals, so that readers could also assess how reliable these identifications were. So it was Dirk Hoyer who did a lot of that work, and he basically he followed the fauna succession as established by von Koenigswald, so with a series of different faunal stages, and he was more focused actually on the fossils themselves to describe really the fossil remains and what were their characteristics, how did species from Java differ from other related species, maybe from the Siwaliks or from China. And he was a bit sloppy, I must say, with the accuracy of the locality data of the fossils in the Dubois collection. So a lot of fossils in the Dubois collection, they have no labels, and during the Second World War, the building was damaged also, so a lot of fossils became mixed up. However, there are clearly fossils that still have the original labels in the Dubois collection. For instance, you have labels that say excavated 3-0, 1 meter below the low water level of the Solo River. So that kind of data was really crucial in what John de Vos and Paul Sondaar did afterwards. So after retirement of Dirk Hoyer, John de Vos took over as the curator of the Dubois collection in Leiden. And together with Paul Sondaar, they started to restudy all the fossils, and they noted that there were inconsistencies in the faunal succession that was erected by von Koenigswald. And what they did was they selected only the fossils with these original labels from Quaternary or from 3-0, and they considered those as the assemblage that came certainly from a single layer, the Hauptknochenschicht at 3-0, or from a 50-meter thick sequence that is exposed near Quaternary. And in doing so, they discovered that it was more likely that Quaternary was slightly younger than 3-0, but because there were clear differences in the assemblage with original labels from 3-0 and those from Quaternary. So, for instance, in 3-0, you have a relatively low number of species. It's mostly Axis Lydeckeri and Duboisia, a small bovid. There's also Stegodon is quite abundant in 3-0, but there is no fossil of an elephas, an elephant, an evolved elephant, among the fossils that have still the original labels from 3-0. There are a few fossils that come from near 3-0. For instance, there's an elephas molar from Nantjar, which is 2 kilometers away from the main excavation site. And for instance, that fossil was assumed to come from 3-0 by Hoyer. But clearly, when you look only at the fossils with labels saying 3-0, then there is no elephas. So it looks like 3-0 is a relatively impoverished fauna. Then in Quaternary fauna, which is actually also an assemblage from a longer time period, whereas Trinil is from a single layer, but Quaternary is from a sequence. But in that Quaternary sequence, you find carnivores like hyena. There is a tapir, and above it called Epileptobos, for the first time appearing in the sequence, and also Elephas fossils. So the Vos and Sondaarde proposed to new stratigraphic scheme, with Trinil being older than Quaternary, and not of the same age as Van Keuniswald had done. And then Nandong is younger than Quaternary. There's no more Duboisia present in Nandong. And a lot of the species, including Stegodon, they differ on a subspecies level. Okay, and then this is a bit, sorry, it's a bit a fuzzy image, but that's all what I could find. So then later on, the Vos and Sondaarde included also some earlier faunal stages, and one was the satyr fauna. And the satyr fauna is based on descriptions by Van der Marel, who studied the hippos and the elephants from a number of excavations at the base of the Caliglaga formation near Bumyayu, along the Chisat. And Van Keuniswald described all the other animals. Now, whereas Van der Marel stated exactly from which excavation each fossil came, Van Keuniswald just lumped everything from that whole sequence. And for those of you who have worked in the area, you know, it's several hundreds meters thick sequence of fluvial deposits. And yeah, fossil here, fossil there, and Van Keuniswald lumped that all together, but by no means was that a clear fauna. And that's also what Sonna and de Vos argue about, that the Chisaat fauna is actually a composite fauna, also needs to be studied, need to be more fossils that are, of which the provenance is exactly known, and also with absolute dates. Okay, and then the Trinil Hauptkoggensticht was estimated to be about 1 million year, Quaternary 800,000 year, Nandong late Pleistocene, and then the Punung fauna from caves in the Gunung Sewu were at that time estimated to be between 125,000 and 60,000 years old, based on similarities with dated caves from West Sumatra. Now, a lot of these ages, they are not based on absolute radiometric dating methods. They were mere estimates. And what you see in this original scheme of the Vos and Sondaarde is that initially in the satyr fauna you have Cynomastodon, which they still call Mastodon bumyaensis, but it is clearly not a mastodon. It's a tetralophodon. It's not a tetralophodon. That was the original name assigned by Van der Marel, because actually the molars, the intermediate molars have 3 ridges instead of 4. Tetralophodon means 4. And besides Tetralophodon, that genus has tusks in the mandible, whereas this species from the lower part of the Caliglaga formation has no tusks, no mandibular tusks. So it's Cynomastodon bumyaensis nowadays. And then slightly higher up in the sequence, Stegodon appears, deer, and there were also hippos present. Because hippos were also in the excavations at the base of the Caliglaga formation in the satyr fauna, in those 5 excavations that were conducted by the Geological Survey at the time. Then Trinil, we still have Stegodon, we have rhino entering, more deer species, more bovids. There's also a very rare macaque. Pigs are present, and I think to the left that is rats and porcupines. And then yeah, I get these weird things here in my screen that something has been updated and I don't want it. Got it? Okay. Then getting rubus fauna, we have a pangolin, and for the first time we have both stegodon and elephas. It's not clear, but these are actually two animals here. A tapir is coming, and there's two rhinos, two species of rhino. Hippo is still there. So over time, you see that there's an increase in the number of species. And then at Nandong, late Pleistocene, we still have stegodon and elephas, and rusa, large deer, tapir, tiger, bovids. And then a clear change occurs in the Punung fauna, and that is Punung fauna has orangutan, and this is missing from all the older sites. And Vos and Sonda interpreted this as the first arrival of rainforest on Java around during the late Pleistocene. Also, what was a first for the Punung fauna is the entry of the modern elephant, Elephas maximus, whereas the older elephants from London and Cullen-Rubus, they were Elephas hisudrindicus. Okay, this is a bit the same scheme, which I plucked from a paper by Zaim, Professor Zaim. It's also largely based on the same scheme. So Chijulang, Kaliglaga, they are actually, it was from Koningswald's schedule, but now in the new schedule, we have satyr fauna, Chisat, Trinil, HK, Hauptknochenschicht, as opposed to fossils from river terraces near Trinil, Cullen-Rubus, Gandam, Punung, and Wajak. So pretty sure that you all heard these names. Now, a couple of years ago, I went to Bumyayo with Mas Unggul and Mas Erik from the Geology Museum. And what we did, we took the geological map and we divided the area in all the different catchment areas of these small little rivers. So here you have the Cipang Soran, Kalipang Soran, you have the Chisat in the middle here. And because we couldn't find any very rich fossil localities, so we thought maybe what we can do is we collect the surface collected fossils from the rivers, because that's where most of the fossils were found. We found a few in situ, but most of them were from these small streams. And then if we are lucky, for instance, if you have this river here, it has a very small catchment area. So if you find a fossil here, you can be pretty sure that it's from the lower part of the Kaliglaga formation. And that lower part of the Kaliglaga formation, that is also the place where the excavations near Satyr here and here at the base of the Kaliglaga formation were conducted in the 1930s. Those excavations with Cinomastodon, hippo, and giant tortoise, so the satyr fauna. And then what we could, we found some stegodon here, so we could actually maybe add stegodon in the lower fauna assemblage from the lower part of the Kaliglaga formation as part of the satyr fauna. It's still 50 meters of sequence, the lower part. So yeah, it's a bit of a lump sum, but that's all the best you can do when there's not much exposure other than in the rivers. And luckily, we found one piece of mandible that is still in the collection at the local museum in Chisari. It was collected by the local Sukri, I think. So, and he showed us where he had found this mandible of Cinomastodon, and that was in this little river here, and you can see that if it was found in this this river, it's this side here, and the catchment area of this little river where it was found is only corresponding with the upper part of the Kali-Glaga formation. So that suggests that this mandible must come from the upper part of the Kali-Glaga formation and not just from the base. And this could mean that actually Cinomastodon continues into much younger sequences, even younger than the satyr fauna of the Bos and Sonda, maybe also in the Cisat fauna. The other way around, we found some rhino fossils in the riverbeds here at the base. I think it's one of these sites here. So we can add rhino also into the lower fauna unit. So this is a bit a summary. So Cinomastodon boomiensis, originally excavated in the 1930s, included in Sondar's satyr fauna present. Then it's also, we also found one isolated molar in the lower part of the Kali-Glaga formation. So yeah, confirming that it's indeed in the lower part, but it's also in the upper zone of the Kali-Glaga formation. Then Stegodon trigonocephalus, we found that also in the lower zone, the lower 100 meter of the Kali-Glaga formation, which was not present in the original excavations at Satyr, and also rhinoceros, and then some turtles, aquatic reptiles, and crocodiles. Then there's still some big question marks. For instance, there is one enigmatic molar of Stegoloxodon indonesicus, which was initially described as Elephas planifrons, but it's really very similar to Stegoloxodon from Sulawesi. And in Sulawesi, we have also skull material and mandibles with lower tusks. So it's a very primitive kind of elephant, and this suggests that it's probably from the lower zone of the Kali-Glaga formation. So if you go back here, it was found reportedly from the Cipanglosoran, which is this river here. But unfortunately, the Cipanglosoran goes through all three intervals of the Kali-Glaga formation. So we still don't know where it belongs, in what part of the Kali-Glaga formation. So yeah, that's some result. There seems to be an overlap between Stegodon and Cinomastodon, but there's still a lot of question marks. Also, a fossil that was described as von Koenigswald as Cervus problematicus. It's a skull cap without a horn or without antler. And that appears to be a Boselaphus, the nilgai from the Indian continent, where the females don't have horns. It's quite primitive kind of antelope. And it's also probably the ancestor of Duboisia santeng, the small antelope from Trinil, Enchedon brubus. So it's likely that a skull of Cervus problematicus, Boselaphus problematicus, also originates from the lower part of the Kali-Glaga formation. Now, another thing I should mention is that in the middle here you have this boundary between the Kali-Glaga formation and the Gintung formation here in yellow. And that's what's called by the geologists the Menger horizon, which contains pumice flows which can be dated. And our colleague Michael Story, he dated the layers from the base of this Menger horizon at around 1.52 million years. So we know that both the Satyr and the Cisat fauna are probably older than 1.5 million years. And then above that, there's also a lot of fossils from the Gintung Formation that would be younger than 1.5 million years, maybe equivalent with Trinil or Cullen-Brubus, but it's and you might also find Homo erectus and or stone tools in that interval. Okay, enough about Bumiayu. Then Punung. It was for a long time also not clear how old it was, until in 2007, Kira Westerway and colleagues, they dated a faunal assemblage from Punung 3, another cave in the same area, which had the same faunal composition: orangutan, gibbon, leaf monkey, sun bear, tapir, all animals that are clearly part of a wet rainforest environment, very similar to the other Punung assemblages that were collected by von Koeniswald earlier. Now they tried to date it, and they concluded that the layer with the fossils, it's a cave Brachia, was between 118 and 128,000 years old, so late Pleistocene, a bit older than that Sondra and de Vos and myself thought at first. And Nandong excavations, also a major publication that came out recently. This is an old image from the excavations at Nandong and a map. They were conducted in 1931 to 1933. And in those excavations, they have still left a remnant where this road went. And and for a very long time, it has been a big debate going on, how old is it? And that's, of course, because there were so many Homo erectus skulls found at Nandong. So Oppenhoort and Koeniswald assumed that it was late Pleistocene. Santa Luca, who described the Homo erectus remains, assumed a middle Pleistocene, and he thought that the skulls were reworked from older layers at Nandong. Then Bartstra in 1988, he was experimenting a little bit with a new method, uranium series dating on bone fragments, and they got very young dates. And as we know now, usually direct uranium series dating, even without modeling, just a bulk sample, gives you at most a minimum age, but it's very inaccurate. There's a very large error range. Then Swisher in 1986, he had a spectacular paper in Nature, I think it was, or Science, that Nandong is only 27 to 46,000 years old, so overlap with modern humans and Homo erectus. And that was based on ESR dates, and then these young dates were strongly disagreed with. Then Indriati et al., in collaboration with Swisher, they did a new attempt to date it, and they come out at 143,000 years, plus or minus 20 or 17,000 years. So there's a lot of things to choose from. Pick your favorite age. Now, then came the study that was published a couple of years ago, led by Jan Rizal, and they used a lot of different dating methods. They re-excavated that remnant of the Nandong terrace near Nandong. They collected fossils. They dated some fossils directly, animals. They didn't find any new hominin remains, but they really made a detailed study of the stratigraphy. At the meantime, in 2007 to 2009, I was involved in excavations at the site Sembungan, where we also collected quite a few fossils and also stone artifacts. But yeah, the fossils were not very indicative. There were not many diagnostic specimens. We knew, well, we could find bubulus paleocarabao and some deer that we could identify, but apart from that, there was no Duboisia or anything like, or hippos. It was quite a limited fauna. But that was also part of this dating effort of the terraces near Nandong and along the Solo River. And basically, so different methods, RED-TL, uranium series combined with ESR dating, uranium series, and then another optical dating method, and also some argon-argon dates on pumice fragments, which are usually, if the pumice is not reworked, the most reliable dates, in my opinion. So, and they found that the Nandong terrace was, well, I think I should go to the next slide. Yeah, so this is all the dates they got from the Nandong terrace, and they modeled what would be the most likely age of the Nandong terrace, and this is the area, fasces C and B, where most of the fossils, at least the fossil hominin skulls, come from, and that is bracketed by ages of 108 to 117,000 years old. So von Koeniswald was not far from the truth when he thought it was late Pleistocene, but certainly not very young late Pleistocene, like 30,000 or 40,000. So how do the new Nandong dates and Punung, Sembungan, and also at Menden, we excavated an elephant skeleton of Elephas isudrindicus, a partial skeleton, which was also dated. And if you plot that against the oxygen isotope curve, then Nandong comes out here during the interglacial period. Cipunduy is a fauna west from Bandung. That was an old sandstone quarry, which is now closed. So unfortunately, there's no more material, but there was clearly Elephas maximus present there in that fauna, and Rhinoceros sandaicus. Punung rainforest fauna, very different from all the other faunas, is just corresponding with this brief interglacial with similar conditions as nowadays during the Holocene. So rainforest in Java. Sembungan seems to be a little bit older. Again, we go back to open woodland or a mixed open woodland habitat, gallery forest maybe. And then Menden, we don't have really a lot of data on. We have only one elephant, no other fossils, but it is Elephas isudrindicus. It's clearly different from Elephas maximus, which is also most likely present in the Punung fauna, but based on fragmentary material. We have to be careful with that. So, yeah, the late Pleistocene is reasonably well covered, but there's still a lot of gaps. Okay, then more recently, there's this wealth of thorough studies carried out at Trinil. This joint project between Leiden University and Bryn, Arcanas, and where they have tried to revisit Trinil, try to interpret all the old images and use all kinds of methods to reconstruct from what the elevation of the boundaries are that you can see on those old pictures. Like, for instance, this example, you probably have heard about this. I can't go into the details. It's quite a complex paper. But for instance, here you can see this is the original photo of the Bergh excavation. And they figured out that there's actually an erosional contact here that cuts all the way down here. It's partly obscured by this rubble, but they have used a lot of photos. And there's another erosional contact here. Now, the layers from which the main, what Dubois called the Hauptknochenschicht, they were also able to reconstruct. There's actually two layers, which they called BBC1 and BBC2. And BBC1 was dated at 830,000 to 770,000 years old. But then there's an hiatus. There's also this erosional contact between the two. And the younger stratum with fossils, vertebrate fossils, is 560 to 380,000. So yeah, I really like this study. It's a very thoroughly done and making maximum use of everything, every piece of information that is available. So they also had access to photos from Oppenort, who was leading the excavation, or he was head of the Geological Survey at the time. And they were in the family archives, and based on all this photographic material, they could also reconstruct where actually the Homo erectus femur came from and the skull cap. And the femur may actually be younger because it's from this younger BBC2 channel than the skull cap. So very impressive study. And yeah, here is a bit of another summary of they also conducted the stratigraphic profiles in the remnants along the Dubois excavation. So here you see again these two different units, BBC1 in pink and BBC2 in yellow. And yeah, what the authors claim is that so the Dubois collection is also not that reliable if you even if you just take fossils with the original labels. Now, I think you shouldn't throw away the baby with the bathwater, and I will show that there is still quite a uniform assemblage from Trinil in the Dubois collection. First of all, there's this chocolate brown color, and they are like the fossils. They sound like porcelain, whereas fossils from younger deposits are often not as good preserved as the ones from Trinil, from the lower levels, the Hauptknochenschicht. So, and this is basically what it comes down to, these new dates for Trinil. So in black is the original Java Biostratigraphy based on the Vos and Sondar. So with Trinil, the Hauptknochenschicht here at 900,000, even though we didn't really have direct dates from that layer, but it was an estimate. And then Kellenbrubus, Fauna, Nandong here, that's okay, pretty well established now. And then Punung, the P. So what happens now is that the Hauptknochenschicht actually seems to be younger than Kellenbrubus fauna. Well, partly older maybe, but partly younger. So it is split into separate faunal units. And yeah, it's a pity. But what I wanted to show you briefly is this is a Stegodon jaw from one of the terraces, the younger layers at Trinil, excavated by the Salenka expedition in 1911 on the other side of the Solo River. And it's a very big mandible. And also there was a skull and enormous tusks in that younger terrace. And I have measured all the Stegodon mandibles and teeth from Trinil, and also from originating from other sites on Java and on other islands. And what I've done is I've measured the transverse distance of the mandibles, and based on the molars, there's 6 molars in a Stegodon mandible during the entire lifespan. They are never at the same time in the mandible, but if you can identify which molar you have and which molar is worn, you can actually divide the whole sequence in dental wear stages, 43 dental wear stages. And those 43 dental wear stages are here at the base of the graph. So here's a newborn individual, and here is a very old individual in which the last molar is totally, completely worn down to the root. There's no enamel left. So very old individuals. And I've done that for several Stegodon species. So the red one is from Sumba. There's one mandible, and we excavated the juvenile mandible. So actually, it follows that the Stegodon from Sumba is the smallest of all the Stegodons in Indonesia. Then a light blue is Stegodon sondari from Flores, from the older sites in Flores, from 1.4 to 1.3 million years old. Stegodon timorensis, slightly smaller again, this purple line, growth curve. And then Stegodon sompuensis from Sulawesi, southwest Sulawesi. Then Stegodon florensis from Soa Basin. And then these black squares are the Stegodons from Trinil in the Dubois collection. Whereas the open squares, they are other Stegodons from other sites in Java. Some correspond with the Trinil growth curve. But as you can see here, there's this one point here, and that is that huge mandible from the younger terrace at Trinil. So I think that if you look at the Stegodon mandibles in the Dubois collection, it's still a homogeneous, a single population assemblage of Stegodon. So, and whether that assemblage is from the lower or the upper BBC level, I don't know at the moment. But it's possible that it's the older BBC layer or the younger BBC layer. But it's still valid data. Okay. One thing to keep in mind is that, hurry up a bit, otherwise it takes too long. As you all know, the Pleistocene sea level fluctuations. I'm always puzzled by why don't we find more rainforest fauna assemblages. And one reason is probably because when there's a rainforest, that's not very good for fossil preservation. You have all these soils rich in humic acids, and bones are just dissolved in the soils. And the only rainforest faunas that we find is in caves, where you have special conditions, more less acid conditions because of limestone. But in open sites like in Sangaran, you would not be likely to find any rainforest fossils. Another thing is that we tend always to compare things with modern situation, the Holocene, but that was only that kind of condition with very high sea level. It was only 10% of the entire Quaternary. So most of the time, large swathes of the Sunda shelf were exposed, probably much drier conditions, and yeah, more open conditions also, more grassy savanna type landscapes. So yeah, concluding about Java, it's still a work under construction, but there's some major breakthroughs, and I would say that one of the things that is largely lacking in Java is a biostratigraphy based on small mammals. And I don't know if there's anybody who will take that task on. It's quite, you have to sieve a lot of sediments, fine-grained sediments. So it's quite labor intensive, and you have to work with a microscope. But like in Europe, most of the European terrestrial biostratigraphy is based primarily on rodents because they evolve so fast, so you can better distinguish successive faunas. Okay, then just briefly a bit about Sulawesi, where we conducted excavations at a site called Talipu here, just west of the Wallene River near Cabenge. Cabenge is here. Now the Cabenge industry that was faced by Van Heekeren. And all these other black dots, they are fossils that I collected during my PhD research in this area. Some from single locations, so yeah, really assemblages, but a lot of them were collected, isolated finds, but many of them still in situ. And so I've made this stratigraphic schedule. This is just show you briefly the excavations at Talipu. We went quite deep. We took samples for optical dating methods, and we did some uranium series dates on fossils. Summarizing, these are those two excavations with overlapping layers. This small hill near Talipu. So the top orange is about 4 meters of fluvial conglomerates that are maximum 140,000 years old. Loads of stone tools in this whole sequence of fluvial deposits, some rounded by fluvial actions, other looking very fresh, so they haven't been transported very far. One fossil of a tooth of a large Anoa type of bovid, presumably the ancestor of Anoa, dated at around 130,000 years. And then in the older part of the sequence, unfortunately, the optical dating method was not possible there because there was quite a high background radiation of gamma rays. So all the minerals were completely saturated. So we couldn't apply that. But some uranium series dates on some of the few fossil remains, including Stegodon, Coelurus, and a crocodile. They were older than 200,000 years. Some of these dates were over 600,000 years, but yeah, it's quite unreliable without additional information, because uranium is very mobile. It can go in or it can out of the fossil, depending on the groundwater condition. So we were very lucky because it was just a 2 by 2 meter pit or a 2 by 1 meter pit. And we found some in situ, clearly stone artifacts, flakes with a beautiful percussion bulb, striking platform in this lower layer here. So we think that hominins stool makers were present around 200,000 years ago on Sulawesi, even though we don't know yet who they were, but unlikely that they were modern humans. So this is a bit of summary of that scheme. So we have the Wallene fauna with Stegaloxodon ceilabensis and Stegodon sompoensis. They are both dwarfed proboscideans. We have giant tortoise, crocodiles, and Bergh, which is the most, it's a pig with enormous upper canines, now extinct. And a rat, Pauramis dominator, just few teeth that we found in one of the excavations there. And that's the species that is still around on Sulawesi. Then in a younger deposit that unconformably overlies the Wallene formation is the Tanrung formation. It's a coastal terrace deposit with a large elephant, a large stegodon, still with a giant tortoise, and also with Bergh. Maybe Babirusa, but that's based on a single molar that looks a bit different from all the Bergh molars. But so I put a question mark there. And then at Talipu, we still find Bergh at around 200,000 years, crocodile and stegodon. A stegodon, we don't know if it's a large or a small-bodied stegodon. Then slightly younger at 130,000, we have this first evidence for an Anoa-like creature, and because of the stone tools, hominins entering Sulawesi. And then after 45,000 modern humans arriving, as evidenced by this beautiful cave art from the Maros karst area. And then still more recent, all this introduction of cattle and other commensal animals that arrive with modern humans. So that is just brief summary. And for Flores, we just had a paper out in 2022 summarizing all our information from the last 10 years or so. Stratigraphy, dating, all these colored lines are colored bands are tephra, volcanic tephras that we can identify based on their geochemistry. And we have dated some of these. We know, for instance, that this yellow band is called the Volosege ignimbrite, which is dated at 1 million year old, based on argon dating. And so if we find that layer back in the Sawa Basin, we know that we are at 1 million year old. And then at one place in the center of the basin here, Tangi Talo, we have a faunal assemblage that is clearly older than 1 million year. And we have also some dates here, argon dates of 1.42, 1.27 million years old. And this fauna is clearly different from all the other younger faunas above that yellow band. There is one at Kobatua here that is probably intermediate between the yellow and the red tephra layer. That's where colleagues from Bryn have been excavating. There's also stone tools there. The oldest stone tools are from Volosege, which is this section here where we have stone tools directly under the 1 million year old layer. But no stone tools at Tangi Talo, even though there's quite a bit of raw material available. There's beautiful chalcedony, large boulders of chalcedony. There's very fine-grained basalt, which was abundantly used in the younger sites in the Sawa Basin for flaking. So I think, and we've excavated here about 100 square meters, so I think hominins were not yet around 1.4 million years ago in Flores. So they must have ended somewhere between 1.4 and 1 million year old. But it's always difficult. Absence of proof is not proof of absence. So yeah, probably can never 100% prove that hominins were not present at Tangi Talo. Anyway, this is the stratigraphy, the final succession on Flores. So we have this old fauna at Tangi Talo with giant tortoise, Stegodon sondari, 1 meter tall Stegodon, a small Quora amboyensis, a small turtle that is still around nowadays, crocodiles, Komodo dragons, and rats, although we don't know which rats. There are also some undiagnostic rats, but rats were already present quite early also, Flores. And then in Liangbua, you see a whole range of rats, and currently Susan Hayes and me, we are studying the rats from the Sawa Basin, this blue band. And we always thought there's not much variability, but we have already identified 5 different species. So yeah, that's very promising. And they're also good environmental indicators. So yeah, big question mark is, why does this fauna become extinct, except Komodo dragons, crocodiles, rats, but the tortoise disappear and the very small Stegodon sondari? Is it hominins arriving? Is it climate change? Unfortunately, we have an hiatus here of about half a million years in which there's no fossil evidence. So still trying to find some rich fossil layers in between, but so far no luck. And the same as in Sulawesi, so during the Holocene, modern people, they introduce a whole lot of animals. The last occurrence of Stegodon is in the Ambua at, I think it's 46,000 years ago, same as the last fossils of Homo floresiensis. We do know that Homo floresiensis was in the Soa Basin. There's a handful of fossils from the site Mataminga, dated at 700,000, sorry. So it's a bit too old here, this image. It should be a bit up. But yeah, it kept me busy for a while. Okay, enough talking from me. I'll stop sharing and I'm happy to answer questions.
Thank you very much, Gert, for your very thorough explanation about the biostratigraphy of not only Java, but also Sulawesi and Flores. So maybe we have questions from our participant here. But while we are waiting, actually I have.
Ask in Indonesian, yeah, and I'll see if I can.
Yeah, of course. So pertanyaan bisa diberikan dalam bahasa Indonesia. Jadi silahkan bila ada yang ingin bertanya menggunakan raise hand atau menulis di kolom chat. So I actually have some questions.
Yeah, well.
So first, what do you take on the discovery of rhinoceros on the lower level of the Bumi Ayu, Cisaat and Satir, between Cisaat and Satir fauna? I mean, Koningswald, I remember he mentioned also the presence of a more archaic form of rhinoceros. It was Acetarium, if I'm not mistaken.
That was from Cisande, a locality called Cisande.
Yeah.
Later, Hoyer thought that it was actually a molar of Rhinoceros sondaicus.
So it's Rhinoceros sondaicus. So they came earlier than we thought.
Yeah. As soon as Java maybe emerged. It could still be a bit later than Cynomastodon and hippos, because that lower interval of the Kali-Glaga formation is still 100 meters thick, so it's quite a bit of time. But somewhere in that lower interval, we have already Stegodon and Rhinoceros sondaicus, and also deer, and then maybe. But yeah, it's still a lot of work to be done to fill in the details, and excavations like they did in the 1930s, that would maybe the way to go. They were quite rich excavations, but I don't know. They were not conducted probably in a very archaeological way. They were paleontological excavations, so maybe a bit take it and break it, as paleontologists often do, and not spit by spit and very carefully. So that would take a long time to collect a large collection of fossils from a single layer. But I hope that answers your question.
Okay, thank you. So I think we have one question here from Benjamin. Maybe you can directly ask the question, Benjamin.
Okay, hello everyone. Can you hear my voice?
Yes.
Hi, Ben.
Okay. Hi, Gert. I want to ask a question because we are talking about the biostratigraphy relating with the Elephas maximus, especially what do you think about the oldest Elephas maximus on Java? And can you more explain anything about that? And second question is the boundary between Elephas maximus and Histodendricus, both from morphology and also from biostratigraphy?
Yeah.
Thank you.
Okay. Thanks. Yeah. Thanks for your question. It's a good question. First, I have to say that a lot of fossil fragments, when you have a single plate or lamellae of an elephant, for instance, like they had in Punung, it's very difficult to distinguish between Elephas histodendricus and Elephas maximus. You need really complete molars, and they are quite rare. So one of the earliest complete molars that I know of comes from this site Cipondui, which was a sand quarry west of Bandung. It's now maybe flooded by the lake, but that was a complete M2, and it had the same number of lamellae and same measurements as Elephas maximus. So I think that's the first proof for the presence of Elephas maximus. And a lot of other fragmentary remains, even from Punung. Yeah, in that paper by Kira Westerway, it's identified as Elephas maximus, as you may recall, then 125,000-year-old. But there is a note saying that I don't fully agree with that identification in that paper, and that's possible. I think it's the material is too fragmentary to make. You can say maybe Elephas confer maximus, but usually it's Elephas species. And that's very frustrating for a lot of archaeologists. They want to know if Elephas maximus is present or not. But you have to be very careful. So, yeah. Coming back to the one from Sambungan, the big skeleton, there we have a skull. And Elephas histodendricus has a clearly different skull type than Elephas maximus. It has this concave frontal, a bit like in Elephas pneumaticus also, although in Elephas pneumaticus it's much more a crest or a ridge directly above the nasal opening, whereas this skull has a lot of similarities with the type specimen of Elephas histodendricus in the Dubois collection, which was besides from a site not far from where we excavated that skeleton in the Menden Terrace along the Solar River. So, yeah, ideally you want to have a complete skull, but skulls they have all this pneumatic bone, and with a little of pressure from sediments, they are crushed, and you don't often find them intact. So that's the problem. Does that answer your question? So the oldest clear evidence was in Cipondui and maybe already in the Punung fauna, but I don't know.
Yeah, I'm just wondering, is that still doable to distinguish between those species from, let's say, a complete postcranial?
Maybe size, because like the Sumatran elephant is the smallest subspecies of Elephas maximus. It has a shoulder height of about 2.5 meters. The males, females a bit smaller. But that's a forest species. And you see also in Africa that the Loxodonta cyclotus, the African forest elephant, is also much smaller than the bush elephant that lives in the savanna. And yeah, but when you have to move through dense vegetation, it's probably not very handy if you have very big tusks and a very big body. So that's probably related to a tropical rainforest. And that's why I think that the Menden Elephas histodendricus was more in an open environment, although Mika, who did some stable isotope analysis, it suggested it was also a browser and not a grazer. So it's a bit of a question mark.
Okay, thank you.
If you can comment on that, Mika.
Yes, sorry. Hello, can you repeat that again?
Yeah, I can hear you.
Yeah, sorry. What was that again? Sorry.
Oh, yeah, we were talking about the Menden, the Blora elephant,
Yeah.
and it's very big, so it might have belonged to a more open savanna-like fauna, but that is in contradiction with the results from your stable isotope analysis of that same individual, I think.
Yeah. Because the stabilized result is more a wet situation, wet condition, rather than open savanna. But probably it's also because there's a mix of vegetation or something like that. Plus, it's the edge is kind of you know like MIS5 or something. If I'm not wrong, yeah.
Yeah, and there's quite a big error range in that age. It was
Right.
optical
So yeah.
age, yeah. But yeah, it's often not as we would like to have.
Yes, exactly.
Still, question marks.
Thank you, Mika. So I think we have another question from YouTube from our YouTube viewers. If I may summarize the question, so it's basically asking about the process of adaptation, and in this case, the island adaptation, like the dwarfism, for example. Do we have any evidence of the transition of the process of the dwarfism?
Like the transition from normal size to dwarf, or when that happens, is that?
Yeah, exactly. That's, I think, the question. What the question is.
Yeah, when it happens, it probably depends on when the first founder population arrives on the island, and then gradually, over time, it's a process of thousands of years and many generations. You get that elephants, for instance, they become smaller, and the idea is on Flores, where we have tried to see if there's a correlation between the size of the island and the body mass of the elephants, the insular elephants. And it seemed that there is a correlation if you only take dwarf stegodon from the tropics, from Indonesia, and so excluding the stegodon from Japan. So because, yeah, if you have a different climate, you have different food availability, whereas if you take all the islands and look at the body size and the size of the island, then there seems to be a rough correlation, except the elephant or the stegodon from Matamena. It seems to be too big for the island, but we know that it arrived somewhere around 900,000 years ago. And if that's the ancestor of the stegodon from Liambua, then we can see over this long time span of maybe 700,000 years, it gradually decreased again in size to the small size that we see in Liambua, the same as the much older stegodon sondari, about a meter tall at the shoulder. And that is actually would fit with the size of the island. So yeah, it takes a long time to get the ideal size, so to say. And the reason why that happens is it's a bit of an equilibrium between the food availability and the population, the size of the population. And if I can imagine that if you have a population of elephants that are breeding on an island, at some time they will eat all the food, and then you get mass starvation, and the smallest individuals would survive, and then so you have this mass death, and then gradually the population increases again until the next time all the food is finished. So that's a bit how the process of dwarfing over many years. So the smaller individuals, they have a higher chance of survival, and that's how this process probably works. But it can take many, many thousands of years. And that was always thought that it could occur in maybe 1,000 years. There were some examples of dwarfed island deer that had dwarfed in 1,000 years. But the dates of that study were not very accurate. And I think you can decrease the body size to a certain size without having to do any genetic modifications in the growth. But then after that, you need mutations or changes in the DNA to facilitate even smaller size. And the most spectacular pygmy elephant is from Sicily, only 60 centimeters tall. It must have been amazing to see these little elephants around. But yeah,
Okay.
I hope that more or less answered the question.
Yes, I think it's very clear
Yeah.
now. So our next question is, I think, both for Gert and for Mika. So it's another question from our YouTube viewer, Amir Hakim. The question is it possible that Stegodon to live in tropical forest?
Yeah, I think it's possible. Actually, in southern Asia, you had Stegodon orientalis, which seems to have been a browser that lived in a forested environment, maybe not tropical, but a densely vegetated environment. It was a browser. And yeah, maybe Mika can better address that question based on her study of all these elephant remains from Indonesia, and then see those changes through time. Or if you want me, I can summarize it. So Mika did this study of yeah, go ahead.
Yep.
Yeah. So
So yeah, hi everyone. I'm Mika here. So I'm sorry that I don't have the PowerPoint at the moment because I'm not in my computer. So actually, based on my study, there are some Stegodon from the mainland, which is Stegodon orientalis, which dated. Well, we still don't have the date yet, but it's from the late Pleistocene. Yeah, approximately from late middle to late Pleistocene, and it has the isotopic value of the animal that lives in the tropical rainforest. But unfortunately, in Indonesia, like in all island in Indonesia, we can hardly find the Stegodon that live in the rainforest, except for mixed forest or open woodland or something like that, but never really rainforest. So that's probably the case for Java, Flores, and Sulawesi only. But in mainland, it's possible. So yeah, I think that's my take for that.
yeah,
Okay,
thank you
thanks.
very much, Mika.
Yeah, and if you're really interested in the research, I can also provide the result. I mean the paper or even my study. So yeah, if that's not enough.
Thank you, Mika, for your answer. So, is there any other question from the Zoom? So maybe we still have a little bit of time left. Maybe we still can have one more question. Or while we are waiting for another question, I would like to ask also to to Van den Bergh. You mentioned that the latest research, the latest excavation, the latest research in Trinil have provided the result of the Trinil fauna, HK Trinil fauna, to be younger than what previously known from Sondar and from John de Vos. So the question is, are we now leaning toward to the more previous biostratigraphic scheme as proposed by Koningswald, that he didn't really distinguish between Kedung Brubus and Trinil fauna?
Okay, thanks for the question. Yeah, I don't think so. They are clearly differences in the fauna. And the new excavations that were conducted in that new studies from those two different layers, they haven't really provided some diagnostic species. So there's not complete diagnostic fossils. So it was just a minor excavation just to prove that there's fossil in that BBC1 and fossils in BBC2, and some of those fossils were used for dating. But it's a lot of unidentifiable bone fragments. So I wouldn't say that they are the same as von Koningswald thought. They are clearly different in age, but maybe not as much difference as we thought initially. And that's why we have all these climatic fluctuations during the Pleistocene. So maybe Trinil, the fauna that I studied, the stegodons that I studied, which are relatively small compared to other stegodons from Java, for instance, Nandong, they were maybe during a period of high sea level stand when Java was an island and stegodon got smaller a bit. Whereas Nandong, it's a very big stegodon, and that was maybe during a phase when Java was connected with the Asian continent, and the same for Kedung Brubus, I think. But the problem is that we need more. Well, paleontologists also always complain we need more fossils.
Okay, thank you very much. I think Ruli has a very interesting question here. Maybe Ruli, you can address the question directly.
You still have time?
Yeah.
Sure.
2 more minutes.
Okay, thank you very much, Pak Gert. Just a quick question. According to you, is it for you the reconstruction about the Pleistocene, the Middle Pleistocene and Late Pleistocene geography or landscape in Java, mostly in Eastern Java, is it more to deciduous forest or a proper, really a dense forest or jungle like we saw now in Sumatra? And the second one, and how about its connection to the primates biodiversity in Java during the at least during the Middle Pleistocene or maybe during the Lower Pleistocene? Yeah. And what is your opinion for the Gigantopithecus from Semedo site?
Okay, yeah, thanks. Very interesting, the Gigantopithecus. There was this recent paper coming out led by Kira Westerway, and have you seen that already? Where they dated all the Gigantopithecus sites in Southeast Asia, or not all, but many sites with different methods. And it seems that they become extinct before modern humans arrive. And I think I haven't even had time to read it thoroughly, but yeah, it's probably more related to the local breakup of the forest into smaller patches. And orangutan continues into the younger layers. I think it happens around 230,000 years ago. But anyway, that's a paper just came out, very interesting. The problem with primates is that they are not very frequent fossils, especially in open sites where you have fluvial deposits, or so they're quite rare. And almost all the primates that we know as fossils, they come from cave sites where they preserve much better. So, and there seem to be some isolated orangutan molars from the Grensbank in Sangiran that may be Middle Pleistocene, early Middle Pleistocene, but they are very rare. And if you look at the Asian continent, almost all the orangutan sites, they are cave sites. There's hardly any open sites. So I think there is a big bias caused by the taphonomy of sites. So you have to be very careful that, okay, the tree-hill fauna didn't have any orangutans. Maybe we just didn't find those fossils, or maybe that's a bad example, because we actually do find a lot of fossils at tree-hill. So preservation must have been really fast and probably related to some kind of volcanic event. But yeah, the other question was if I thought that tropical rainforest was spread out all the way to East Java. I don't think so, but maybe during the previous interglacial, it may have extended a little bit further than it does in modern circumstances. So now it's mostly restricted to West Java, but maybe in the past it has moved a bit further east. But I'm not sure. I haven't read any recent papers on that kind of studies, maybe with palynology or other environmental reconstructions. But what do you think, Ruli?
I have no idea about that. That's why I address the question. But thank you very much for your question. And Kira papers, maybe just a few days ago, I think.
Yeah, it was in Nature.
Published. Yeah, it was in Nature. Yeah, interesting article.
Thank you very much. I think we are a little bit running out of time for our talk show here. We still have another speaker, who will give the closing remark. He is Unggul Prastio, is from Museum Geologi Bandung. But before, I would like to thank you for your time, Gert, and thank you for your presentation and all of the answers that you give to our audience. And I'm looking forward for more discussion with you about the biostratigraphy. So thank you very much for your time.
Has been my pleasure.
So Unggul, it's the mic is yours.
Okay, thank you, Pak Mirza and guys. So I feel not proper to make a close statement because as I know in here, so many experts in here. So yeah, it's my honor to make a closing statement, but I will try my best. So I follow this seminar from 1 o'clock until now, and thank you for Mr. Gert that we have insight about the history of the biostratigraphy in Java or in archipelago of Indonesia in common. And then it's interesting because you try to explain from the history and then from the technical, and then also follow to the from the data that recently still problem and still need to be discussed. So I have some note in here based on the discussion this afternoon. So I wrote about the the biostratigraphy based on the fossil can explain about the migration and the evolution. So some of the speaker in here ask about the evolution, especially based on the proboscis fossil. We know that Mika also studied about that based on the geochemical and can explain about the evolution of the and paleo environment based on the geochemical of the fossil teeth fossil. So it's a new insight in here in the paleontological aspect. And then I also have noticed about the okay so the discussion going to discuss the changing of the paleo and beginning isolated island and then evolve to become more truly island, and then sometimes connecting with the mainland in the Asia. So we know that until now we recognize 3 corridor migration from the west is from the India and then Malaya and then through to Java, and then also from the second corridor from the China mainland and then through the south and then come to the west of Indonesia, and then the third corridor is from the Philippines and then going to the Sulawesi and then until end in the Flores. So I think this discussion is interesting because so many new data that got bring in this seminar. So we know that some biostratigraphy must be updated in here, especially in Java biostratigraphy. He saw about some new data that likes probably Stegodon trigonocephalus comes more early than what we know right now. So yeah, we still need more fossil, like Gert said, that we need to explore more. So I think that's my my close statement. So we still need more fossil and still need to digging. So thank you.
Thank you very much, Unggul, for your closing remark. I return this microphone to our Master of Ceremony.
Thank you, Mas Ami. So we already have a really beautiful presentation from Pak Gert. We are arriving to the end of the talk show today. I hope everybody enjoyed the talk show, and please follow our social media to get an update from CPAS, from Instagram, from Facebook, and from YouTube to get an update from the researcher in CPAS about our current research, current issues in Indonesian archaeology and prehistory studies. And for the last, we would like to thanks to Pak Gert. Thank you very much for joining us. And Pak Unggul also for the closing remarks, beautiful closing remarks. Mbak Mika and Benjamin Perwira, our colleagues, and all of you from the participant and from the YouTube channel who already viewing us and participating in our talk show. And see you in another talk show from CPAS next month. Thank you.
Okay, thank you, and see you tomorrow.
Thank you very much. See you tomorrow. See you tomorrow also, Mika. Thank you very much, everybody. I hope you enjoy. Terima kasih semuanya.